Okay, let's start. So the next lecture is glomerular filtration and renal hemodynamics. So this is just the overview of the kidney function. Again, we start with filtering a lot of fluid in the glomeruli and reabsorbing most of it. And the next slide kind of shows just the big picture. I think this is missing from your handout, so you can take slightly notes, or it's going to be on the recording. So basically what happens at filtration is that the blood enters the glomerular capillary system, and the glomerular capillaries here have very high hydrostatic pressure. And this very high hydrostatic pressure is going to push through roughly 20% of the plasma into this space called the Bowman capsule, from where the fluid is going to go to the tubular system. And from the first approximation, just to very much simplifying it, the primary filtrate is basically protein and cell-free plasma. So proteins are large, cells are even larger. Those are not filtered, but everything else is filtered. Sodium, glucose, calcium, whatever is in the plasma. It's basically protein and cell-free plasma, which is driven from these glomerular capillaries into the Bowman capsule and then into the tubuli by mainly the high hydrostatic pressure in the glomerular capillaries. We'll go into the details of what else is determining it, but this is the first approximation, simple view of what's going on. Because the plasma, the filtrate is protein-free, that means that the protein concentration in the blood leaving the glomerular capillaries will be higher than in the blood coming in. And the importance of this will be that the efferent arterioles, which is the arteriole coming out of the glomerular capillaries, is going to divide into capillaries one more time, the secondary capillary system, which is called the peritubular capillaries. And it's important to note that the peritubular capillaries have higher protein concentration than the glomerular capillaries, and that will gain some importance in the Starling forces that dictate movement of fluid both in the glomerular capillaries and here the reabsorption from the tubular cells eventually will enter these peritubular capillaries, and the high protein concentration there will be a driving force for fluid movement back into the capillaries. So the next slide shows essentially the same thing. It's a cross section. Again, you can see that the blood goes in to the glomerular capillary system through the afferent arteriole. It divides into capillaries, and from these capillaries, it will be basically filtered into the Bowman capsule, from where it goes to the tubular system. And the blood leaves the glomerular capillary system through the efferent arteriole. One more thing that was also shown in the previous one I didn't emphasize is that these tubuli, this tubular system will go down to the deep medulla, then it comes back, and when it comes back, in this structure called the macula densa, it will touch the same glomerulus. This will gain some importance later, as you will see. Now, this is a slightly different plot of different representation in more 3D. You can see the same thing. Blood comes in the afferent arteriole, leaves the glomerular capillaries through the efferent arteriole. Here is the glomerular capillary loop. Here is where filtration happens. Again, showing the macula densa coming back to the same tubular system. And if you zoom in, you can see that the inside of the capillary, this is the filtration surface from inside of capillary through this thing called the podocyte layer. You can see these interesting looking cells that cover from the inside of the Bowman capsule, the capillaries. These are called podocytes. And the reason why these podocytes are very important, because as I said, the glomerular capillaries have very high hydrostatic pressure compared to systemic capillaries. And capillaries are not made to withstand high hydrostatic pressure. They have very thin walls. And basically what you have here, these capillaries are basically covered by these interdigitating finger-like processes of this other cell type called the podocytes. So these basically support these glomerular capillaries so they don't explode from the high hydrostatic pressure. And the podocytes together with the capillary membranes will be the filtration surface through which the filtration actually happens. So if you zoom in even deeper to this, this thing and take an actual electron microscope picture, you can see that this is the capillary side. You can see that the capillary wall is has these large holes. It's called fenestrated endothelium. So basically the idea is that since these holes are so large, these are very unlikely to be any kind of barrier for filtration. What basically holds back the proteins is the next two layers, which is the basal lamina, these glycoprotein and proteoglycan structure, the basal lamina, and in between the podocytes processes, food processes, you can see these little diaphragm structures. These two together are the two surfaces that basically make it difficult for proteins to cross this membrane. And the importance of that is that it turns out that the proteins that make this diaphragm between the food processes have proteins which, if they're mutated, people end up with proteinuria and kidney damage because they don't these structures don't hold back proteins anymore. So let's go to the details. So I said the filtrate is protein-free plasma. Basically, what determines what is filtered and what not is largely size. Anything that's below 7 kD in molecular weight is freely filterable, and anything that's larger than 70 kD is not filtered. And in between 7,000 and 70,000 Daltons, it's partially filtered. And what determines how much is filtered in this intermediate range is the charge of the compounds. And the reason it's important is because albumin, which is the most important, quantitatively the most important protein in your blood plasma, is in the range that it could be to some extent filtered, but it's not really filtered too much because it's negatively charged. And basically, this negative charge prevents it essentially from filtering only to a very, very small extent. And even that very small amount that is filtered is usually endocytosed. So we normally don't really have proteins in our urine. So any urinary dipstick, if there's protein in your urine, that means there's something wrong with your kidneys. So the normal glomerular filtration rate is very high. It's 120 milliliters per minute. If you calculate it to a day, 175, 180 liters a day, it's an enormous amount of fluid. Roughly 20% of plasma on average is filtered. If you divide the glomerular filtration rate with the renal plasma flow, it tells you that 0.2 or 20% of the plasma is filtered. And why is that? If you think about it, one reason is that it's pretty genuine that anything that enters your blood and hydrophilic enough to be filtered, and if you don't have any mechanism for the kidney to reabsorb it, it's just gonna go. It goes to your urine, and the more you filter, the more stuff you can get rid of. So in a way, it's advantageous to have a high filtration rate because it basically makes the kidney very efficient in getting rid of things that you don't need. The lower your glomerular filtration rate, the lower the maximum clearance you can achieve for anything that enters your blood that you don't need it. Of course, the trade-off of this is that you have to have very efficient energy-consuming mechanisms to reabsorb much of that fluid. Otherwise, you would have to drink tens of liters a day, which is not the case usually. And One more thing to note is that this system is very much over-secured, so you don't need such a high glomerular filtration rate. Most people, if they're born with one kidney, they're usually okay. You can donate your kidney to your relative if they need one. You can live perfectly normally with one kidney. So the 2 kidneys with the 180 liters is not something that you normally need. But of course, it's always good to have plenty of reserve. So it's better to have 2 kidneys than 1 overall. You're really only considered to be clinically in renal failure if your GFR is below 15 milliliters per minute. And again, the normal is 120. So let's come back just to revisit inulin clearance one more time. So the glomerular filtration rate is one of the most important kidney parameters that it can be measured and it can be estimated. If you want to measure it precisely, then you can use the clearance principle. This is just an additional way of deducing why GFR is the same as inulin clearance. And you can look at it in a way that if something is filtered but neither reabsorbed nor secreted, then the amount in the urine should be equal to the amount filtered. And so the amount in the urine is U times V. The amount filtered is GFR times P. And if you make these 2 quantities equal and you solve it to GFR, then you get basically the clearance equation. This is another way to explain why inulin clearance is the same as glomerular filtration rate. Now, in real life, very, very infrequently, clinicians measure inulin clearance or clearance of anything that's similar to inulin. The reason for that is because the plasma concentration is a moving target. So if somebody gets just 1 injection of inulin, the inulin concentration will go down pretty quickly. And to estimate the urine flow, you need to collect urine for an extended period of time. So the only way this measurement actually works, if somebody is in the hospital and gets continuous infusion of inulin or something equivalent for a long time and collect urine for a long time, and that's really, really cumbersome. So very rarely it is done. To avoid the need to inject anything to people, scientists and clinicians look for things that can be used instead, something that's endogenous, and creatinine is such compound. Creatinine is a byproduct of muscle metabolism. It's formed in a nearly constant rate. It's freely filtered and not reabsorbed. Unfortunately, it's secreted a little bit, but the secretion is not significant enough to prevent the usage of creatinine as an estimation for the GFR, but it's something it's good to keep in mind that it's still to some extent happens. So if you want to be really, really precise, you can measure the clearance of creatinine. For that, still, you need to collect urine for like 10 or better yet 24 hours, which is still a pretty cumbersome thing to do. And so as an end result, as a simple measurement, most of the time, if you go to a physical exam to your primary care physician, they routinely measure plasma creatinine concentrations. And the idea is if somebody is in a kidney failure, their plasma creatinine concentration would go up because the clearance goes down and eventually creatinine will accumulate in your blood. So creatinine is a widely used kidney parameter used in really everyday clinical practice to basically estimate if you have any problem with your kidneys. This slide just shows that plasma creatinine, the relationship between plasma creatinine and GFR is not as perfect as it would be if it wasn't secreted. So these are the straight line is calculated values. This how it would look if it wasn't secreted, but it's secreted a little bit, so it deviates. And the take-home message from this is that creatinine in the normal GFR range is not a super sensitive measure for GFR. If your GFR drops from 120 to 80, the change is so small that it's really hard to reliably tell that you have any issue. When it really shoots up is at or close to the kidney failure range when it becomes really high. if your GFR drops below a certain level. So let's go to the Starling forces that determine glomerular filtration rate. So the rate of filtration equals the filtration coefficient times the net filtration pressure. The filtration coefficient basically depends on the permeability and the surface area. Those are really hard to measure, but if you have, say, kidney fibrosis, that would increase this or decrease this filtration coefficient, but it's not something that can really very exactly be measured. So, but what can be quantified is the Starling forces, which determine the net filtration pressure. And just like in systemic capillaries, the fluid movement to the capillary wall depends on the hydrostatic pressure and the oncotic pressure. And so the profiltration forces are the hydrostatic pressure in the glomerular capillaries. And the anti-filtration factors are the hydrostatic pressure in the Bowman capsule. If, for example, you have your ureters constricted by some sort of either a tumor or, let's say, a kidney stone, then a back pressure can develop and basically increase your Bowman capsule pressure, and that could stop filtration. But normally, it's not something that actually changes too much. And you also have the oncotic pressure in the glomerular capillaries that you do have protein, which acts against filtration. You don't have much protein in the Bowman capsule, so it's basically 3 pressures. So again, the major driving force, the reason why the kidney capillaries, the glomerular capillaries filter a lot more than systemic capillaries is the high hydrostatic pressure. And the reason for that is the special circulation of the kidney that normally in any circulatory system for any organ, the highest resistance to blood flow is the arterioles, the little vessels before the capillaries. And if you have the highest resistance, you have the highest pressure drop. And normally in systemic capillaries, the pressure drop is very high at the arterioles, so the hydrostatic pressure in the capillary is very low. But in the kidneys, you have 2 consecutive arterioles, so you have a certain pressure drop in the first system, but you have another large pressure drop in the second one. And basically, the glomerular capillaries are stuck in between 2 arterial systems. The hydrostatic pressure is high, therefore it serves as a significant force for filtration. So these are the glomerular capillaries. And also the second capillary loop, on the other hand, has very low hydrostatic pressure. These are the peritubular capillaries. These are important also because when you reabsorb the fluid, the large amount of fluid from the tubular system, that fluid still has to enter the peritubular capillaries to go back to the general circulation. And this low hydrostatic pressure is an important factor in enabling this. So the fact that the kidney circulation has 2 different arterioles, it allows this organ to selectively constrict one or the other capillaries, or at least preferentially, and the hemodynamic consequences will be different. So if you think about it, if you constrict the afferent arteriole that's before the glomerular capillaries, what happens if you constrict that? The pressure behind that constriction will drop. Think of a garden hose. If you have your garden hose, the water is flowing, you step on it, and the water flow at the end of the hose will go down because the pressure goes down. On the other hand, if you constrict the efferent arterioles, right, after the glomerular capillaries, what happens, they will develop this back pressure. If you think of the garden hose, you step on it, and before where you constricted it, the hose will bulge. If you puncture a hole, it will sprinkle up. So this back pressure can increase the glomerular capillary pressure. So again, afferent constriction decreases glomerular capillary pressure, efferent will increase it. And of course, if you constrict both, it depends which one you constricted more, what the effect will be. If you use this principle to plot the consequences of efferent arterial constriction or increased resistance, if you plot the effect on the renal plasma flow, the glomerular capillary pressure, and the glomerular filtration rate, you will see that as you increase the arterial resistance, all of these three parameters will decrease. Quite logical, because if you increase the resistance, the plasma flow will go down. Since the constriction is before the glomerular capillaries, glomerular capillary pressure goes down, and therefore GFR will decrease quite dramatically, because both the flow decreased, there's less to filter from, plus the pressure decreased also. This is afferent constriction. If you constrict the efferent arteriole, the consequence will be somewhat different, because what happens here is the renal plasma flow will decrease. If you increase resistance, flow will decrease no matter what, but your glomerular capillary pressure will increase, as we also discussed in the previous slide. And as a consequence, your glomerular filtration rate will have this biphasic effect. It will first increase, because at the beginning, your the glomerular capillary pressure increase will dominate. But of course, if you constrict it a lot more, then at one point, the renal plasma flow decrease will dominate, and eventually it could also decrease. But for much of the discussions we will have in these lectures, for a moderate efferent arterial resistance, the GFR usually goes up a little bit. So the bottom line is that the afferent resistance is a more efficient regulator of GFR. So if you want to regulate the glomerular filtration rate, you can play with the resistance of the afferent arteriole. On the other hand, if you consider the efferent arteriole, what's going to happen is GFR goes up to some extent, but the plasma flow goes down, which means your filtration fraction will increase quite a bit. So you filter a larger fraction of the fluid if you increase the efferent arteriole, because your flow goes down, but you filter, if anything, a little more. So your filtration fraction will go up. This will have importance for angiotensin too. So keep this in mind for now. So, so the regulation of the renal plasma flow and the glomerular filtration rate can be divided into extrinsic and intrinsic factors. The intrinsic factors are things that do not depend on either innervation or hormonal regulation. They would work in an experimentally removed kidney. So if you take an animal, take out the kidney, just perfuse it artificially, the intrinsic mechanisms will still work. So the intrinsic regulation of the RPF and the GFR is called autoregulation. So what you can see here is an increase in the renal arterial pressure. And what is plotted here is the GFR, the renal blood flow, and the vascular resistance. So you can see that normally by Ohm's law, if you increase the pressure, the flow will increase. But what you can see here is that let's look at the middle one first. What you can see here is that after a certain increase in the pressure, if you increase it further, there's not going to be much more increase in the flow. This is called autoregulation. You probably had this from circulation. Certain organs have quite pronounced autoregulation. Which one has the most pronounced other than the kidney? The brain. It's important for the brain, basically, because if you have a large spike in blood pressure, you want to protect the brain. So you constrict, because otherwise your blood vessels can blow up. With the kidney, you want to protect against filtering too much, because 180 liters is a lot to deal with. if your blood pressure goes up and you filter 240 liters, then at one point you might reach the limit of the reabsorption capability, and you don't want to lose insane amounts of fluid. So basically, the autoregulation is important, and of course, the whole idea is that you keep the flow more or less constant in order to keep the glomerular filtration rate constant. And the way it's achieved is an increase in the afferent arterial resistance, because constricting the afferent arteriole is a more efficient way to regulate your glomerular filtration rate. And the way it goes, it's basically one of the mechanisms, a simple built-in stretch sensing mechanism in the arterioles. So if they stretch, then you're going to have these stretch-activated ion channels that open, let calcium in, and basically constrict the vessels. This happens in almost all vessels, especially so in the brain. But the kidney has an additional mechanism, which is called the tubuloglomerular feedback. And this is where the importance of the macula densa, this recurrent reconnection of later parts of the nephron with the glomerulus, this is where it becomes important. So basically what happens here is this macula densa, which is part of the distal tubular system, it essentially monitors the flow of the filtrate. And if the flow is higher than normal, then there's going to be higher sodium chloride delivery to the macula densa, and that's going to be sensed by this sodium potassium 2-chloride transporter. And basically what happens is you have too much flow here, it just sends a signal to the efferent arteriole, they're essentially touching each other, sends a signal to the efferent arteriole to constrict and decrease the filtration. And this is a beautiful mechanism because this is local. So basically what happens is the previous mechanism happens generally, but what happens, this is individualized to each and every nephron. The idea behind it is that biology is variable. Sometimes weird things happen. And so if a nephron goes crazy and filters twice as much as the other nephrons, chances are that particular nephron won't be able to deal with the extra filtered fluid. So what happens is basically it evens out the individual differences between how much each nephron filters, because any nephron that just stochastically starts to filter like crazy, immediately there's a built-in mechanism to slow down that particular nephron to avoid overflowing of the system. So this dual mechanism, the general stretch-induced constriction and this tubular-glomerular feedback ensures that the glomerular filtration rate remains relatively stable, independent of systemic blood pressure and independent of individual variabilities or increases in single nephron GFR. So this was the intrinsic mechanism, where the idea was to keep it constant, not to depend on blood pressure too much and to even out individual changes in filtration of single nephrons. What you also have is extrinsic regulation, and these are vasoconstrictors, namely the renin-angiotensin II system and the sympathetic nervous activity. And you have vasodilators like ANP and prostaglandins. So the extrinsic regulation, the most important one is the renin-angiotensin II system. So the renin-angiotensin II system is the major salt conserving system or sodium chloride conserving system. We will discuss in even more detail on the next lecture. One of the very important effects will be for angiotensin II to stimulate aldosterone. But angiotensin II also have a very important effect on renal hemodynamics. And what angiotensin II does, it's a vasoconstrictor, and it constricts the efferent arterioles more than it does the afferent arterioles. And the consequence of that will be is that the renal plasma flow will go down, but the glomerular filtration rate will go either up or don't change too much. And the end result is that the filtration fraction goes up. And if the filtration fraction goes up, that means what's going to happen to the protein concentration in the peritubular capillaries if you increase the filtration fraction? It's going to go up, down? Up, yes. So if the filtration factor is higher, that means there's going to be more proteins in the peritubular capillaries. And the importance of that will be, it's going to be easier for fluid that was reabsorbed to move back into the general circulation. We'll reiterate this concept a couple of more times, but this efferent arterial constriction is important because it increases the oncotic pressure at the end in the peritubular capillaries, making it easier to reabsorb large quantities of fluid. And fluid usually follows sodium because that's the most important osmotic compound. So it's important for sodium balance to be able to reabsorb as much fluid as possible. So the sympathetic nervous system, you have learned about this in detail at the cardiovascular system. If the blood pressure drops, the baroreceptors in the carotid sinuses and the aortic arterioles sense it, and the sympathetic activity will go up, norepinephrine being the neurotransmitter. And what happens is it vasoconstricts both the afferent and efferent arterioles. And both this one and the effect of angiotensin II, basically the vasoconstriction drops the renal plasma flow, and that has, of course, effects on the ability to reabsorb sodium. But also you have to remember that the renal blood flow is very, very high. So if your blood pressure drops, you can decrease the renal plasma flow without decreasing your renal function and basically redistribute blood to your more acutely vital organs, which is basically your brains and your muscles. Your kidneys are vital organs. You die without them, there's no question, but you can essentially turn off your kidneys for a day and you're going to be okay. So basically decreasing the renal plasma flow is one mechanism of maintaining your general blood pressure. Of course, you can't decrease it to too low, because if you don't perfuse your kidneys, they're going to die of ischemia, and that actually does happen sometimes. But basically the concept is that there's a significant wiggle room to change the renal plasma flow to help other organs receive more blood, because acutely the large plasma flow is not that vital for the kidney function. You can decrease it a little bit. So the sympathetic also has this crosstalk with the renin-angiotensin II system. It will also stimulate the renin release. So angiotensin II will go up. We'll discuss in more detail how angiotensin II is regulated. So the end result will be that if you have a moderate sympathetic activity, it might induce a small decrease in GFR. But if you have extreme sympathetic activity, you can have a very significant decrease in both renal plasma flow and GFR. And if, let's say, somebody is in shock, the kidneys can get so low perfusion, and the sympathetic induced constriction makes it even worse, that you can get ischemic kidney damage, especially that the renal medulla has very low circulation, as we will discuss later. And so the extrinsic regulation of the GFR, you also have vasodilators, the most important one being the atrial natriuretic peptide. It increases the renal plasma flow and increases the glomerular filtration rate. So basically what AMP does, it's the exact opposite of the renin-angiotensin II system. So if you have an increase in the extracellular fluid volume, your low pressure system, which is mainly your atria, will sense it. If you have high extracellular fluid volume, your venous system will mostly accommodate it. So if your venous system is dilated, which includes your atria, that's going to basically send a signal to your kidneys to dispose sodium to decrease your volume. And the AMP, atrial natriuretic peptide, is part of this system. So it dilates both the efferent and efferent arterioles. The renal plasma flow will go up, the GFR will go up, and it's also a systemic vasodilator. So even though in principle it increases the renal perfusion, but if there's too much of an increase, your blood pressure can drop, and that could counteract this effect. As we will discuss in the next lecture, it also inhibits renin secretion and decreases sodium reabsorption. And finally, you have prostaglandins, which are prostaglandin E2 and I2. These are vasodilators. These are local substances made in the kidney. Basically, what they do is they are secreted in response to angiotensin II and increased sympathetic activity to essentially counteract this vasodilation, to make sure that the sympathetic and angiotensin II wouldn't completely stop the kidney. And the reason to know this is that non-steroid anti-inflammatory drugs like ibuprofen, Motrin, these compounds, they block the formation of these prostaglandins. And if somebody has a low kidney perfusion or heavily dehydrated, it could be dangerous to take these drugs, because basically these vasodilators can be the last little thing that keeps the kidney being perfused. And if you block it, then you can basically stop kidney perfusion. So this is something to keep in mind. And this lecture went a little faster than I thought, so we can have a 10-minute break, and we will still have plenty of time to finish the last one. Thank you.